So to get going then, this first 45 minutes, and it's probably less than that now, is to really sort of define some of the problems. And we come back in the breakout session to see if we can move towards solutions or paths forward. And so if you look at the Google Doc, which is actually, I realize, hard to do on my small screen together with the Zoom, the sort of first thing we decided as we were looking at this topic of clinical metadata is that we really don't know how to define it. So Sarah and Melissa and I tried amongst ourselves to define what we mean by that. And we thought that that's actually maybe just worth a quick set of time here in this room amongst people. And I'm hoping that those of you who are participating by video might jump in and give us some ideas. And we'll try to document these as we go. So I will begin this. And in many senses, in terms of forging a larger network, we noticed that there are other groups working on metadata standards in these breakout sessions. There are other groups working on sample collections and biospecimens. And in many ways, we actually think that all of these areas interact. So none of us were entirely clear on what the scope of clinical metadata was and what that really meant. So to give you an idea of what it means to me from GTEx, the word clinical to me has this sort of clinical physician, patient, hospital setting. And thanks so much. And coming from a perspective of healthy individuals and donor sampling, that doesn't really translate that well to me. But some of the things that I think encompass clinical metadata include things that are related to the donor or the participant or the patient, depending on what you call them, but also in my world, some things that would be assigned to the sample, but are particularly relevant in sort of this single cell area where part of those collections or early type of collections matter to the sample quality. So to give you my list of the type of things that I think of in the scope of clinical metadata, there are obvious things like information about the donor or the individual, demographic, such as patient age, sex, BMI, other things, ethnicity, things like that. Perhaps information about the consent or whether there are constraints on the consent from individual to study to study. And then in the context of deceased donors, we have a whole suite of things that are sort of donor related that do affect downstream analyses that include the actual cause of death. Was it motor vehicle accident? Was it coming off a ventilator in a hospital? What was the actual cause of death? And then what was leading up to the cause of death? Because that may have medical or clinical implications. The circumstances of death, as I said, was the body refrigerated? Was there a ventilator? The timing of death, and in particular related to the ischemic time, not just for the individual, the timing of death, but also then every sample you collect from the individual. We also get medical history, but in the context of healthy individuals, it's often just documented by word of mouth interview as opposed to coming from a medical record. Smoking and alcohol, which may again be interview based. And then other things such as the tissue sample, really where did we sample tissues from, which feeds into this whole common coordinate framework, but really comes right at the point where you get a donor. Pathology, histology, sample preparation, and preservation. So those latter ones become very sample focused, and they may be part of the things that feed into the sample collection group that comes later on, either today or tomorrow. But to me, all of those feed into our suite of things that we know make a big difference when we analyze downstream data. So I'm going to stop there and actually open the room to participants and see what people think this scope of metadata is. Is it entirely clinical, hospital-based? Is it entirely donor-based, or does it include a slightly broader subset, like I said? So it would be great to spend some time just to get some ideas that we can jot down and follow up later. And I don't know if Sarah or Melissa, you want to jump in with an opinion next, and then we'll open it up to people.
sure, just something to add, as Melissa kind of showed you as she scrolled down, is we put a couple comments and welcome people to comment on the bottom. So if you scroll all the way to the bottom of the Google document, or if you use kind of the indexing, you'll see that we put together some suggestions based on just what Kristin has just walked through. But we welcome people to suggest things there. We're going to put things in the question notes so they're participating in accordance, but we can also add more comments there as well.
Okay, so active participants, can we get some feedback, ideas, brainstorming? Just let's get this going. What do people think of as the scope of clinical metadata?
Hi, I can start. Asaf wrote them here from Dana-Farber. Very nicely presented, and taking from our experience in the human tumor network, it would be very helpful to go in a structure that first we address basic information that will be relevant to all the samples, and then go into specific samples and specific collections. For example, if it's GTEx or fresh samples or preserved samples. So that would be my 2 cents for a start.
So I think, Asaf, you're leading us into the next topic too, which is to define some core, suggestion of defining some core fields, but it does also sound like you include some bleed into information both about the donor and pure medical / clinical, as well as sample related. So this link between donor and sample, is that correct?
Yeah, so maybe it's how might we structure the metadata collection?
Okay. Can we have some more contributions and ideas?
Yeah, hi, Mark Kalishka, Johns Hopkins. I'm a cardiovascular pathologist, and I agree that defining what we take and how we take it is really a critical piece of this. Just to completely agree with what Kristin and Asaf said, but starting at the tissue itself, where's the tissue coming from? How long has it been devitalized if it's a surgical case versus an autopsy case? And getting the metadata around the tissue is very important for quality of the data, and then be able to correlate that sample with other samples that come from other resources is a really important piece. And getting that consistent from group to group, I think, is a key, hopefully a key function of this meeting and this discussion right now.
Can I speak to that briefly from CBTM in the UK? In theory, depending on how you collect the tissue or where the tissue is collected from and who's doing it, quite often it's, if you say up front, it can be kind of recorded. So with CBTM, because we collect tissues from deceased organ donors, it's the same team of us, so we have a preset protocol. So we know exactly which region we constantly take the same tissue sample from, unless obviously the researchers come back and tell us they want some variation on it. If you're getting samples from through kind of biopsies during medical procedures, because each time the surgical team might be different, keeping that consistent location is going to be the bigger challenge in those scenarios. So it might be worth looking at whether there's routes to keep those consistent components, or whether what you're taking is a consistent sample.
So I think you're saying something that we clearly have a similar perspective here, as does Mark and Asaf, which is that this concept of the clinical metadata to us extends absolutely to that sample collection piece of the biospecimen. And it is critical. We know it's critical for downstream analyses, because the subtle variation there can make a big difference. And really documenting that is key. But in any case, in terms of this definition of what do we think is clinical metadata, it seems like we're all agreeing on this scope that includes the donor and the donor through to that initial setting in the sample collection. And I think it's really key, these pieces are key, because it's really what helps to tie us into a common coordinate framework, whether we have some of that information both in a setting where we can control, like deceased donors, and where we can't control, like biopsies or surgical settings and so on. But there are others in the room. So what do others think? What is the scope of clinical metadata here? What do you think it should include? And I guess later this afternoon, we may touch on what do we think we can control, what do we need to standardize, but we'll introduce that next. So anyone else on the scope of clinical metadata?
Kristin, may I just interject for one moment, which is to say, everyone is most welcome to type into the chat window. That's absolutely fine, and it's a useful thing to do. But I'd also like to encourage you to add thoughts and questions into the Google Doc, just to make sure that nothing gets lost. It's and also Sean is doing a lovely job in capturing people's thoughts. You might want to check that there are no nuances which you would like to add. The wonderful thing about Google Docs is you can indeed be anonymous pumpkin, as I just noticed somebody was. So feel free to have it open and add thoughts and annotations as you go along. And back to you.
Yes,
Hi, sorry,
please do,
just a quick
because I'm not
here.
actually looking at the chats while I'm going, I'm afraid. So go on.
Yeah, so I'm trying not to identify myself as an active scriber, but you are saying that then anybody can go in and add that, because I don't want to confuse the official scriber.
I think the suggestion was that as thoughts are being noted down and you can actually see them in real time, if you felt you wanted to add something to it, you can. And also one of the nice things in Google Docs is you can sort of highlight a bit and add a comment to it, which I often find is a useful way to do that. So we don't want to encourage all out chaos, just minor bits of chaos. I just would like to make sure that people feel that their content is being captured.
So could you just give us a bit of guidance on where in that document you think we can put our stuff without messing anybody? In the green sections?
The green section is where a lot of this is being captured. That's the thought. And without wishing to derail the process too far, it was just, I'm noticing as people are capturing the comments that are being said out loud, just keep an eye on it. And if there's anything you want to add to it, you can. Back to you.
Okay, anyone else have some ideas on sort of what the scope of clinical metadata means to you and should mean in the studies we're doing?
Hey, Kirsten.
Go ahead.
Hi, it's Alexander Misharin, Northwestern Chicago. I had a pleasure to work with some of the people who did meta-analysis for the research data for AC2 and TMPRSS22 expression. And I think that was a very interesting case because working with Lisa Sikam and Malte from Fabentai's group, we've pulled the data from different data sets, and that was exactly the case when we started tapping into very simple questions about metadata, such as age of the subjects, what the tissues were taken from, what was the ischemia period between the tissue collection and processing. And I think even smoking status was really very interesting question because everyone tracks it differently, whether it's active smoker or, smoker is person who ever smoked in the past. So I actually think that these use cases, where we ask a very specific question and try to apply it to data sets generated by different groups, that these use cases can actually identify the core sets of variables that we need to capture. And again, I understand that the work of HCA is focusing on the healthy tissues, but bringing the disease context, I think this really helps actually to capture the right questions. So maybe after this meeting, we can pull out a couple use cases, maybe lung or kidney. I think those communities actively contributed. And just really ask, how can we normalize and integrate and compare the metadata between different centers?
I actually, thank you so much, Alexander. I think that's fabulous. Sarah, Melissa, and I thought that the best way to approach this topic was through use cases. And of course, that's a very topical and very recent use case. So I think that that's a great idea. And you're going to have an idea of what the scope of some of the metadata is out there. The challenge I have, and maybe it's really for not now, but as we go forward, is that nobody really likes harmonizing metadata. It's a horrible thing to do, and it's also quite hard to sort of set standards that go globally, which is some of our next topics. But it might be nice as a group to decide on what are some fields that people do get routinely, and are there ways as a community that we could suggest people at least harmonize or present their own, so that these meta-analyses, we're more able to do these meta-analyses downstream, because it is very, very difficult, and nobody likes going back, and nobody's willing to fund massive efforts to go back and harmonize all these old data sets either. But it's very, very worthwhile, and I think looking through a use case, especially in the single cell area amongst our community, the couple you mentioned will be very worthwhile. So I know we have a time limit, and Sarah and Melissa wanted to go onto some of the other topics, but I just wanted to ask, is there anyone else who thinks we haven't captured what clinical metadata might mean to them in their work, or the challenges with it? So does anyone else want to speak up so we can summarize any other viewpoints?
So this is Sean, and so just to reference, my perspective is coming from the Human Tumor Atlas, and I think looking in the notes that Sarah may have noted some of this below, but for HTAN, both the longitudinal data and also spatial information about where the samples are collected from are important components for the metadata and building the atlas.
Yes, exactly, and for us to feed into that that framework, that common coordinate framework.
And Kristin, I see that Anita Umesh has also put a comment in our chat box. I don't know if you have enough screen space to see
I have
that,
not been able to see comments while I'm moderating.
Anita, would you like to unmute yourself if you want to explain that to the group?
Oh, hi. Can you hear me?
Hey,
yes, we can. Hear you.
Well, which part? So I have a little bit of experience working with aggregating clinical data sets from different sources, such as public data sets and clinical trials, things like this, and linking it with omics level information from my past jobs I worked at a startup called NextBio. I don't know if that's familiar to some people, but so what we did was in aggregating all these clinical, this information from these different data sources, we had, I don't know which part you wanted me to explain or speak to, but this definition of sample is very important to sort of, I think, clarify, because between the people that came from the biology side versus the folks from the bioinformatics side to the people who are from the software engineering side, we all had a different understanding of what we were referring to as a sample. So some of the database people that I worked with, the software engineers, a record in their tables was a sample. And that was a big disconnect between the bioinformatics folks who were referring to an assay, a result as a sample, whereas the biomedical folks like myself, we were referring to the biospecimen. That makes sense. So just as a caution, it might be helpful to go back to first principles and define the simple things. And also to make a distinction between what is coming out of the subject level information versus the biospecimen level information, because the metadata, the structuring and hierarchical structure of the information that you collect from a subject that goes down to the biospecimen, that goes down to the assay level, to even the platform in which a certain, whether it's RNA or DNA is analyzed becomes important.
I just like, this is Ian 4, I'd just like to add some comments to that. I mean, you've taken that even broader than I was thinking about it. I mean, the idea, right, where a sample, from a statistical point of view, is that thing which we extract from the population and which we do numbers on, right? So as a standard error is a statistic about your sample. But a sample here could be 100 patients or 1,000 patients, depending. But distinguishing that, as you say, from the biological sample. But where I was focused with it, and I put a comment in the document about this. I mean, we've had the discussions where we're talking about, is this a sample or a specimen or an aliquot or a segment? You know, we had situations where people dissect a particular physical piece of biological material and dissect it into different pieces. And the discussions around the precise meaning of which one is an aliquot and which one's a specimen. And people have different hierarchies that they turn up different ways for that. I think the discussions around there being a right answer to that are not productive. And that what we need is the flexible way to allow those hierarchies to be recorded rather than everyone harmonizing on a particular nomenclature for it. I mean, and some of my background with this, or immediate background, is looking at the HTAN project and seeing that the depth of hierarchies and the complexity of the hierarchies for that are going to be way more complicated than anything we dealt with in things like GTEx or TCGA.
And I thank you so much, both of you. I couldn't agree with you more, Ian. And I think we're going to face that level of complexity in the aliquot and everything else as we go downstream in the projects we're working with here. But I'm going to make the general comment, which is one that Melissa and Sarah and I discussed when we started this, that we felt like talking about clinical metadata and trying to define that was not in the context of a common coordinate framework or the downstream analyses is very hard to do. And in the end, we think we don't necessarily need just a working group going forward in this area, but one that links to these other groups for the reasons you've just articulated, that a donor goes to a sample, goes to an aliquot, goes to an assay, and they're interrelated. And so stopping at one doesn't make sense. That said,
I like your term common coordinate framework, and could you say more about what that is? It's possibly addressing what in...
There is actually a working group session on it in the course of this meeting. So I'd encourage you to attend that. But the goal there is, and Mark sort of alluded to it earlier, the general goal is to have a coordinate system across the body to try to tie where samples are taken from so that we can link data, which proves to be very difficult depending on the particular organ, the age, and so on. But that's the idea, to try to at least spatially and physically tie samples to a location that can tie them back to analyses. But as I say, there's a specific session on that, although what we do here, particularly since we're at the front end of dealing with having a donor or a patient and getting a sample, clearly feeds into that. But Sarah, Melissa, do you want to, shall we bring this back down to what we think is the scope of some things we can define here and move on to the next session, which was going to be sort of, is there anything we can define as a core? I've lost track of what our agenda was at this point.
Yeah, so maybe if Melissa, since you're sharing, if you scroll back to the top of the main topic, it is, I think we've highlighted a number of these things here, and if you look below in the Google document, you'll certainly see we've broken these questions out one by one, which we've now kind of started to address. I think we could talk about the core. I think a couple people have posted information about the HCA, for example, that we could talk about there. And I know that somebody wanted to kind of spend some time on the naming conventions, and maybe that's part of this linking the individual patient to the biospecimen to any of the downstream samples that might be generated. But I wanted to also gauge, are there other of these topics that we've listed here, whether it's review, how different groups review data or data sharing or any of these other kind of generating new standards that have any precedent, or if not, since we only have another 15 minutes in this session, although we can continue, I think, into some of the second part of this session, how we should prioritize them. If anybody has any opinions, but I think certainly going to subtopic 1 and we can kind of expand on this is the next place to go. And then maybe it's jumping down to the naming convention.
I'm going to jump in just a little bit and say, I think the subtopic 1, obviously, we'd already started to move into that. And subtopic 2 is relevant too. It's really, you know, what we need in the context of when we're doing cellular studies, the human cell atlas, the hub map, spatial assays. So several of the speakers have already alluded to that as a key thing. And it's really why those of us are sort of leaning in this general area where clinical metadata also defines some of these sample collection parameters. So I think those 2 subtopics are ones that we've clearly jumped in on. But I don't want to take anything away from the other ones if people want to continue. So I'll turn it back to you, Sarah. And if anyone else wants to add any opinions.
This is Melissa. I just noticed in the chat there was a comment from Marion. I'm sorry, I can't. Shadbolt, that you're working with some other groups. I wonder if you might just be willing to unmute and say which group that you're working with on these metadata standards so that we might think about who to collaborate with.
Yeah, sure. So my name is Marion. I'm based at the EMBL EBI in Cambridge, and we're part of the data coordination platform team for the HCA. And so I work with contributors who are want to contribute their data to the HCA DCP. And we are also working on defining the metadata standards for those contributions. So we define our metadata standard as JSON schema, and we're constantly trying to get feedback from our community on the kinds of fields that they want to capture and what's important to understand their experiments. And we also, with that bearing in mind that what we want to achieve at the end of the day with the HCA is have a large corpus of data that's interoperable so that it has all metadata that can be integrated so that you can really get a whole picture of the human biology at a single cell level. And we think metadata is really important to that. So, yeah.
Yeah, so I think that's really helpful to kind of clarify some of the things that you mentioned. Do you provide kind of these folks that want to add kind of maybe additional variables that you hadn't included, do you provide them with a kind of format? We try to have these baseline variables on everyone as we go into talking about these clinical core versus kind of these other maybe newer concepts that might be more specific to an assay or an experiment or something like that.
Yeah, so currently we have our fields, and we have certain core fields that are required. So we say, like, you must tell us certain things about your experiment. And then other fields are optional. We're kind of toying with the idea of having an idea of like a recommended field. So it's not strictly required, but we'd kind of like to have it. In terms of adding new fields, that's generally, we don't have the ability to kind of, I know some resources you can just kind of create your own custom field, but we kind of don't really do that. It's more like if we get feedback that this is a field that would be useful for a certain contributor, we can then kind of have a discussion about, you know, how important is it? Is it important to the wider community? And then we would think about adding it to our schema. So we're not like super flexible, and we end up, we kind of capture it within more like a free text field instead of a strict field. But then if we get enough of a sense that it's an important field, then we would look at editing our schema, if that makes sense.
Yeah, I think that makes some sense. So Melissa, could I just scroll down to maybe the clinical metadata, and maybe we could start adding more comments here as we spend a few minutes talking about this. I agree, Kristin, we have really kind of gone into some of these questions, but I don't know if some of these questions might bring out some more things that we have or wanted to discuss further. So one is this idea of having some set of standards. I can speak for the HTAN, for example. We utilize the standards, the GDC standards. I think that we're utilized in the TCGA for some of our kind of baseline clinical metadata. And we've worked, the DCC as part of the HTAN has suggested, along with kind of different investigators that help to lead some of that effort to what might be required or optional. And I think groups are now kind of, so there's that baseline, and this maybe ties into the second subtopic as well, as there's also additional levels that are outside of that baseline or core that are going to be kind of developed as time continues, that are more maybe specific to cohorts and certain disease types, or other kind of levels of clinical data that become more specific. And maybe that's similar to what you're having people kind of suggest and have free fields of entry. So I don't know, we could talk about kind of other data sets other than the HCA and GDC maybe as standards, but I don't know, Kristin, if you wanted to talk about any specific standards or try to get to answers around any more of these. I think there's pretty much an agreement, yes, there should be some core set of standards. And I think there's been mention now about kind of normal versus disease and the really importance of how the samples are taken and things like that. And so I don't know if other people have comments there.
I think, yeah, I don't know. I guess I don't know how much time we've got left, but I think I really liked the idea of looking at what were some of the available metadata and how diverse were they in the use cases. And that's probably something we can follow up in the afternoon session. So I guess the two examples that were given this morning were the lung data and the kidney data. And these are all data sets that are being produced in, globally right now. Everyone's producing data rapidly, but in very different circumstances and settings. And we touched on some where they're actually from a discarded tissue or a deceased tissue, some where they're from a diseased tissue, some where they're biopsies. So we have very different amounts of information. And it would be really good to sort of think about how we could use those use cases to see what we got, what we didn't. And then there are the more structured projects such as GTEx, HTAN, others who've decided on what a set of core data are, and are there anything we can learn from those as key core metadata that you'd want. But how, have those big studies done them in such a way that even they're easy to harmonize or not? And then there's issues with studies that, don't have maybe a blood sample or something like that, where you get very limited data. So I guess what are the core limited sets of data that we'd really like to try to get, and what are the issues with formatting even when we get them?
Kristin, let me just jump in because you were asking about time. We have about 4 minutes left, and then through the magic of Zoom, I suspect we will be rather unceremoniously pulled back into plenary. And if that doesn't work, then you just go back to the welcome page and click the plenary button, or click again on the link that would have been sent to you when you registered. My suggestion is, when we come back for the second part of the session, it seems as though it would be most useful for us all to come back into plenary, and then a first part of our discussion will be whether people want to go into the subtopic breakouts. Does that seem reasonable?
Yes. I think we maybe, well, I guess maybe let's come back into a group session together and decide if there are a couple of breakouts we want to go into, or whether we want to continue as a group. The problem we have is that some of our subtopics are just so interrelated.
Yes,
I think.
I do. I understand. So just as general guidance to everyone who is here now, this will be the room that we come back to at our next breakout session, which there's going to be the Francis Collins presentation, and then a very brief sort of orientation again from my colleagues, and then we come back for the second part of this after a stretch break.
Great. So I think in our last 2 minutes here, I think maybe kind of there aren't maybe specifics about each of these. I think you're right. We've really kind of covered many of the different components here. Is just to say that maybe we'll start the next session talking about kind of these naming conventions, and maybe this gets to the component that'll talk about kind of how do we relate patient to biospecimen to sample, or if it's not patient, species, or kind of wherever the sample type starts, but also maybe also discuss some of these, how we're managing multiple entries. So a patient may have multiple samples taken at once. I think this is true for both HTAN and GTEC, for example. And as Sean brought up, I think something that we're gonna be challenged with in the HTAN is this concept of time points. So there are a number of the studies are longitudinal in nature, as well as the spatial component, which I think is something that I'm really interested to see how others are kind of addressing this, because this is something we continue to be challenged in thinking about in the HTAN. So I might leave it at that, unless Melissa or Kristin, you wanted to add anything else just in this 1 minute before we all get pulled back.
Yes, thank you. So just as a reminder, the purpose of the second breakout is for us to start to propose how we might move forward with the work. We don't have to come up with solutions today, but as we continue to define some of the topics that people would like to work on, that the goal of that next session is to try to outline some of the next steps.
I guess maybe I'll let either Sarah or Melissa sort of lead this area so that I don't talk for another 75 minutes. But if I'm just summarizing where I see that we were, it seemed like the group as a whole agreed that this area of clinical metadata, especially with respect to the types of studies we're talking about, spans both a sort of donor, participant, subject, and a sample. So it might be worth, as we said, just going to discuss the perhaps the naming conventions next. But clearly, there's you know, this group agrees that it spans those areas, and it seems to me that going forward, looking at a current couple of use cases for what metadata there are, what exists, what people collect when you start to pull these projects together that span both that classical donor, clinical, and the sample collection, physical state and space, and you know, what are we missing? What is it really key to define as core things that we'd like to get or like to encourage people to get, or if they have it in a format, to sort of standardize some format going in. And then to tie that back to groups like the HCA, DCC, I guess the HubMap one, these projects that are starting to ingest data, what are the kinds of things they're already ingesting that span these fields, and you know, how can we standardize and really get a core set that's key? And you know, maybe Mark can chime in. He's done a lot of analyses of what happens when you miss data and how it can lead you astray. But I'll let one of the other two lead this session, although I'm happy to jump in.
Sarah, do you want to lead off?
Sure. Yeah, so do you want to scroll back up? We'll just spend a few minutes here, and then I agree, I really want to get into kind of how do we solve some of this, what kind of work we could do together to really push that forward. And so if you actually scroll down to maybe subtopic number 3, I think the same information might be there. And so this is somebody else. I think, Ian, you mentioned this is something that we should certainly talk about, so I welcome you to chime in as well. I could speak a little bit to what we've been thinking about in the HTAN just to give some context, but I'm really interested to what others are doing as well. And so within the HTAN, as we've mentioned already, there will be a number of donors. Most are live, but not necessarily all. We have samples coming from patients that are either retrospectively, so found in banks, or prospectively being collected, and in many cases longitudinally collected over time. And so there's going to be, for each participant, we'll say, there's going to be a number of samples, there's data entries, clinical data that may change over time. Or there's multiple data points. So these are some things that we're trying to tackle as we think of the naming convention. So at this point, the ID schema that we have been using is that there is a patient ID that is kind of named by the center. So again, a little bit of background about HTAN. There's 10 centers and 2 additional pilot programs that are represented within the HTAN. And I think if you went to some of the presentations in advance, you might have known more of this, but basically each center will have their own ID scheme, and then they can identify their patients accordingly. And then from there, every biospecimen gets an ID. And as biospecimens are broken out into what we might call samples, or their aliquots, or their blood samples, or they might have some different kind of levels. The naming schema is the same throughout. And so a biospecimen ID where that could be, and then from there, there's a metadata point that suggests what exactly is that a subsetted sample, is it an aliquot or an analyte that's been isolated, and so on and so forth. And for each of those kind of sub-biospecimen IDs, they're linked back to the patient ID and often associated with a time point. And how we're doing that is using index dates throughout that. The spatial component, as I mentioned, that's the part we're struggling a little bit with of how, and curious to hear today and maybe throughout some of the other sessions, how other groups are kind of tackling that, if you are tackling it at all. And what we mean by spatial components, and the way that I see it, but I welcome other people to suggest otherwise, is if you're thinking about, for me, a lung cancer specimen where there might be an area of tumor, there might be an area of normal, and there might be an area of pre-invasive disease. And so in our case, we might be isolating via laser microdissection or highlighting regions of interest for protein-based staining, is how do we kind of label those appropriately? What is the appropriate origin? And so on and so forth. And so I think that depending on your area, you might come at it from one direction or another, and what you see is the true origin, but that's something we're struggling with. So I'll call it a challenge. I don't think we have an answer just yet, but I welcome people's thoughts there. So I'll just put those out there as some thoughts, but I'm curious to what other groups are doing.
Sarah, could you repeat your challenge so I make sure I capture it?
Yeah, so I think the challenge is really defining how we identify like the origin, or how we appropriately, not index isn't maybe the right word, but how we provide information that would allow us to link back, okay, this pre-invasive area within a tumor section is at this location, opposed to an area of tumor might be in this other location. And so the way kind of machine learning expert might do is that they set an origin, and then they use kind of distance from the origin to identify that. So some sort of XY or XYZ, depending on the type of tissue, but we're working to figure out to define that. And I'm curious if others are struggling with that. And so I think these are related to the naming conventions, but maybe a little bit of an adjacent.
So Sarah, this is Ian. You noted that I was interested in this topic, so I throw in a couple of comments. One thing was, I think we've got some experiences over, I think certainly NCI, but I think many people have been involved in some of the projects connected with NCI. So I'm thinking of GTEx, and I'm thinking of TCGA over the last, I'm going to say 10 years or more, and have gone through some of the situations that you're describing. And it just came off GA4GH meeting last week, and was at the same GA4GH equivalent meeting at Hingston last year, and talked to Tony Burdett about some issues related to this, and I think Marion, who's on here, is out of Tony's group, and I was just trying to make the connections between Tony and the discussion here, and he mentioned Marion and Laura Clark, I think. But I Paul, you know, to bring this to crystallize this, I mean, and I think Tony's comment here echoes my comment, which is hopefully we got away from bake all the semantics into the sample name. And I think with TCGA, certainly, you know, there's an experience over that time about the problems that occurred where one went through this increasingly long sample identifier, which was trying to do two things. One, it was trying to identify the sample, but it was also trying to tell you by looking at the identifier, you knew that it was what it was, and therefore it had some semantic meaning. So you could look at that identifier and say, and it would capture, for example, the kind of things that you're saying. This, I mean, and looking at some of the HTAN projects, they're capturing, for example, different sections of a tumor, for example, something that in glioblastoma, I think it was identifying something that has a high level of neoplastic cellularity. It wasn't actually, I'd have to go back to look at the document. But simplifying this down, I mean, the question of the naming and the identifiers, I mean, what TCGA did was ending up going with a non-committal identifier that tells you nothing about the sample, basically. And the linkage between the samples was dealt with in another way, which is in what's been coming up in the discussion in the Google document today, is this question of provenance. And so trying to do that all through the name, I think, is what both Tony and I and others we could bring to the table to talk about this. Is doing it through the name is not the way to do it.
Yeah, so I think the kind of DCC within the HTAN agrees with you, and they are not using that approach. And so for each patient ID, I just put an example in the sheet. This is what a patient ID would be. A biospecimen sample might have a similar thing, but it's not going to extend past that. So this 001 might be the patient, and then it would be _001, and then it would just be the biospecimen ID, for example. And this is as long as the ID's naming will go. And so this patient 1 might have 1 through 50 different biospecimen IDs based on kind of, that would then have metadata that would delineate time or what type of sample and so on and so forth.
So when you referred earlier to there being aliquots or blood samples that came from that level, they're still going to have something that identifies them. So had you thought about what that would be?
So at the moment, all of that information will be captured as metadata. So that's information, so each site would upload, for example, on every sample that they'd have a patient information that they might attribute clinical information that's kind of cross-sectional, but then they might have several biospecimen IDs where they would have the biospecimen ID written, and then it would have a next kind of information to be filled out would be the patient ID that it was associated with. And so that there is a structure to tie back any biospecimens to a particular patient or other biospecimens from that patient.
So I think that's good in that, you know, these longer and longer identifiers don't emerge, but it's still the case that besides the metadata, one would need an ID. I mean, the system doesn't work at an engineering level, or it's very hard to make it work at an engineering level, if those things those lower levels don't have IDs. And we can call them IDs rather than names, if we want to settle on name being the thing that humans can consume. But though they're going to need identifiers, and perhaps we can get away with them being hidden to everybody. But those identifiers, I think, are an essential ingredient to linking into the metadata that you're describing.
Yeah, if I understand it, yeah. So for each biospecimen information, regardless of if it's a true biospecimen, it's the whole sample, or it's a subset of sample, any information tied to that.
But to give a specific example, if we got an aliquot, and we know that the hierarchies for H10 are going to be quite deep, and we could be 2 levels of depth down from the level that you are referring to here. But there may be some piece of processing or some attribute of that thing, and it's a real thing. It could be a slide, for example, upon which some immunohistochemistry was done. There are going to be some pieces of information about how that was processed. And you're going to capture those attributes, but you've got to have to hang those attributes off something. And the ID that I'm referring to would be the thing that you hang those attributes off. So if there was some, let's say it was something to do with fluorescence, and there was some attribute to do with the illumination that needed to be recorded on that slide to capture that image, you need an ID for that slide.
Yes, I absolutely agree. And that would be the, I could show you an example. Is it all right if I share? Are people interested in how this works in the HN and what it would look like? I can quickly show you an example, if that's helpful.
This is Melissa. I just wanted to jump in and just say, I just want to make sure that we capture all the questions today and then put on a list of post work that we would go through elaborating on all the details that you all are diving into here. I think that in fairness to everybody, that we make sure that we spend the time capturing all the opportunities and the problems that we need to solve, and then how we want to go about moving them forward.
I think we're getting a little into the weeds right here. I mean, that's like a pretty deep dive into one thing. One of my overarching questions is who's coordinating. I think there's a lot of great ideas. A lot of people are doing things, but at what level? And I know I feel like it's probably under Laura Clark, who should be organizing everyone to get on the same page, but this group's doing their thing, this group's doing their thing, and it's like if we could just take the best ideas that already exist today, bring it together, and then require all the groups to use these best practices, we would be ahead of the game rather than everyone have to try and figure things out. And I'm just figuring out, I'm hoping at this meeting, we can establish some level of coordination among groups that already have really good practices that we can all take advantage of.
The other thing, and I'll add to Mark because I agree, which is a lot of this work is distributed efforts all by different groups. I saw that Jonathan left a couple of messages in the chat about how HubMap's doing, and it would be good to hear from him too. But I don't know that we can sort of push or solve something going forward. And a lot of what we're talking about gets back to having an ID that is really where that ID is identifying something, but it's the metadata around it that's telling us what that is. And so I think that's really the provenance of this group, and maybe the thing to do is to understand what some groups' best practices are, to make a suggestion that an ID that I think we all agree with does not have information in it, but then to check, does the metadata surrounding that ID, is there some logical check we could put in place where you can link that ID to the metadata you need and find out where you are at the aliquot level, at the sub-dissected level, at the original biospecimen level, or where it is. So is that metadata around it sufficient in every case to really get us where we need to?
This is Jonathan.
So it sounds like that would be something to capture on what we want to come out of this breakout session of next steps, is to capture how to tackle those topics. Sorry, somebody else is trying to speak. Go ahead.
This is Jonathan, because I was called out. I figure I might as well speak. But I have two quick comments. One is in terms of coordination, the HubMap PIs had a specific discussion about this in preparation for this meeting to figure out how we can get useful action items to come from this meeting with all the discussion that's going to go on, and then we just go back home doing about what we're doing. So to Mark's comment about how do we actually move that forward, I think I can speak to some degree for the HubMap PIs to say that our feeling is that we need to identify specific actionable things, whether they're even just between two groups or, you know, taking an idea from here and implementing it even on our own, but hopefully, you know, with at least two different groups working on something together, and not try to solve everything in a massive governance process, but choose things where someone picks up and says, hey, this is something that I've been thinking about, looks like they've already done it, or vice versa, looks like nobody's done this, maybe a couple of us can get together and try to do that thing. So we're hoping many of those small specific items with very small groups can be the outcome from this, rather than a specific governance process across consortium, because we're concerned about the speed that may move. So that's one comment in regard to your comment, Mark, of what we're hoping comes out of this are specific actionable collaborations that do small things but move the inter-consortium collaborations forward. I think there is a CFDE process which is within the Common Fund program, which to move Common Fund programs to do the same thing has taken a similar attitude and seems to be making forward progress now. So that was on that. And then on the specifics, this ID business and metadata, just because I offered some of the ideas that we were doing in HubMap, I'll just say that we've been very specific to separate these different concerns. The reference data, which is the ontologies and terminologies and spatial coordinates that are canonical, from the provenance and from the querying of all this stuff with metadata. So we bring the metadata in and annotate the provenance graph with it, but for search, we actually then export all that stuff into Elasticsearch. And so none of this has been deployed to the community yet. So it's all in baking right now. And, you know, we'll have a big coming out of all the things that HubMap has been doing sometime a few months from now. And so it's a funny position for us to be solving a bunch of these things without a lot to show for it, but that's where we are. And so I would say that the overall comment in that regard, I think for this group, is where Ian was talking about the IDs being stuffed into, or the metadata being stuffed into an ID, to me is only the smallest tip of the iceberg of a kind of problem that we've fallen into to try and do everything through schema and pre-baked across the board. So where there's reference data, that can be reference data. Where there's provenance, it can be provenance. Where there's metadata, it can annotate provenance items, and using multiple different technologies to bring that together with APIs is the approach we've taken, really in a microservices type of approach. So I think we can make a lot of progress on what clinical metadata we're interested in this group in the next couple of days, and then with projects after that. I think, of course, the provenance and the other things are incredibly closely tied to how you operationalize that. But I think if we try to eat that whole elephant at once, we end up mixing technologies and processes. And so I was hoping that this group would focus more on really the clinical metadata, and whether we're taking an EHR-oriented approach in which there's a lot of experience with clinical data, or there's some reason to take a different approach when moving that data into the biological sphere. So I'll stop talking there, not to dominate the conversation, but I'm hoping that we'll work on some of those things.
Thank you very much, Jonathan. And just to speak to your last point and Kristin's point about the different groups in this session, in these 2 days working on related topics, I think that from my own perspective, one of the things that we can do is look to where we want to collaborate with these other groups. Because, for example, the group that's working on metadata schemas, you know, it's directly related to this decision about what kinds of data are we working with, and where there are already well-established standards, and where we need to really contribute and build new standards, or build use cases for existing standards that haven't been brought to the table yet. So I just wanted to note that as something that perhaps we can put in one of our next steps to come out of this meeting, is to figure out, have a plan for who to collaborate, which groups to collaborate with to move this forward.
So, I mean, in relation to that, I mean, I have a question for Jonathan. I mean, the separation of concerns that you were talking about, I think makes sense. And I see the clinical data separating off in the way that you describe as making sense. My question is then, and this would, you know, help to identify a group to collaborate with, where do you think the right place is for the provenance discussion, and where is that taking place, Jonathan?
I think because it's everywhere, and so I'm not really sure the answer to that. Yeah. We have a particular approach that we're deeply committed to. We've been collecting our samples for the last, 6 months or so on that. So everything has these IDs. We have a working model of it. Literally is working with all these different entities. So I feel like that's an area where we're, despite the newcomers, we're unusually ahead in HubMap. And so we'd like to share that and interact with folks and show people what we're doing and see whether folks can use that or create isomorphic implementations of their own that can generate the same kinds of data from whatever they've created. So I felt that that was somewhat separate from here. I don't know where that's happening within these next couple of days, but it doesn't have to be somewhere. We can establish that it's needed, and in the gallery and other places towards the end, establish an interest group and move it forward.
I think it's important to do that because you're saying it's everywhere, and I get why it's everywhere. The problem is if it's everywhere, it's almost nowhere. And the reason it's everywhere, I mean, as I looked at the agenda and thinking, where's this issue going to come up? It's because it affects everything. I mean, there are groups talking about sample attributes and how to collect them and so on. And this clearly affects them. But as a core issue, it seems to me that it makes sense to bring it together somewhere, but it is something that is used by many.
Yeah, this is Jonathan Himmelfarb. I'll just point out that in the breakout groups this afternoon, there's a session on tissue collection and processing. And a lot of the provenance type issues will get discussed in those breakouts.
Okay. Also, I think we've come to kind of just breaking these into two parts, and certainly some kind of important information to think about in the context of what we've been talking about kind of in the early part of this morning. I don't know if anybody else has anything else to add here. If not, I think we can probably move down to breakout session 2, where it seems like there's an interest in focusing maybe a little bit more on the actual clinical metadata, but also maybe highlighting, especially how do we move this, anything that we decide on today forward. And so we've put a couple of questions in here, I think that have evolved over the last day, this morning. But again, I welcome people to think of other questions that we maybe should be hitting on. I don't know if Melissa, you want to move to the bottom, and if you want to lead this, start off this breakout session, or this part of the breakout, I guess.
Sorry, I was on mute. I just wanted to get back here, levels of metadata. Okay, I just want to make sure I know where we are. So we also thought that this topic of the review process, the actual process of how we're going to come to agreement, I think is important to discuss. We have a lot of different consortia and working, and we have some people who seem to have the broader view. So what if you have some thoughts on where some of the opportunities are for working more collaboratively or bringing everybody's work together? That was the purpose of this topic. And if there's a process in particular or what some of the opportunities are for creating a process for actually reviewing and coming to agreement will be important. And if this is not interesting, we could also think about moving to the purpose of this whole session, which is to start to think about what would be the next steps after this meeting to start moving some of these topics forward.
Melissa, this is Sean. Could you say just a bit more about what you think we should come to agreement on in the reviewing metadata? What like core metadata
So, well, Sarah, you can help me out since you brought this forward. The idea is that we need to have a common process for coming to consensus on what the metadata are or what the data elements are that we want to standardize around, and how to bring everyone together to be able to get enough input to make sure that these will be robust standards.
Yeah, I agree. I think this last question, I mean, this is what we were just talking about previously, is really how do we work across, not just within our groups or consortia, but really maybe across consortia, and what are the things, I think, defining kind of those things as part of today. But are there good methods that are working in your consortia for review processes? Are we trying to establish kind of these core set, and how do we go about that? Who are the players that need to be involved, and how to maybe move that forward is part of the questions, and how this is maybe now becoming relevant. Like when we proposed this, we were thinking kind of maybe having discussions around this if this was a question of how the review process goes, but I think more so this last question is what's become kind of most importance today.
I just want to jump in, am I on mute? No. And say that Anita and Jonathan just had a useful exchange on the chat where Anita was asking, is this group trying to look into the practices for collecting information or for the tabulation in a database for the purpose of analytics, and that the two are not necessarily compatible? And I think I agree with Jonathan's answer that they're intertwined and they're both key. And maybe we're involved in both. I think the discussions this morning clearly, brought up the fact that we're trying to make sure we actually collect some of these core metadata pieces so that they're not missing. And maybe that should be a goal of this group, which is, can we identify some of those? Can we encourage the collection of those? But then the second, because this is distributed, diverse efforts, the second really plays into this as well, which is how we tabulate these in databases, how we make sure that they're standardized for analytics. And I think both matter. Do either of you two want to comment?
Yes, I mean, I can say that from working in the clinical research space, we have standards that we use for collecting for clinical trials where we have different representations essentially of the same information in a collection standard, a reporting and tabulation standard, and an analysis standard, because the use cases are different. It's the same information, but we might ask the question differently from the way that we tabulate it in a data storage, or that the way that we need to represent it for analysis. So I think that being able to represent the information consistently across use cases, it will be key to being able to automate later and be able to have clear visibility into the data through its entire chain of collection, through storage, through analysis. So there is precedent for doing that. I think it's a great question and a great observation. And it will be important for this group to be able to address those needs as well.
Can I ask a question as well? Someone had brought up the electronic medical record, which is interesting because we're all mostly moving to EMRs now. In the United States, a lot of places are using Epic as the main EMR, and we're able to generate searches and pull data out of EMRs, or Epic in particular. Would it make sense if groups, and I don't know how many places are using Epic in particular, although it's very common now, that this group or similar could make a search that could be built at your institution, that could be shared to other institutions, that everyone is pulling the same data fields out of the EMR together? Is that something that could be done?
In connection to that, I mean, the story goes, I mean, and you could substitute any EMR vendor in here. So I'll substitute Epic in. As if you've seen one implementation of Epic, you've seen one implementation of Epic. And because those systems get customized at each installation, the fact that they're using the same EMR system doesn't necessarily mean they're compatible. Have we got past that in any way?
So this is Jonathan. I can address this. I mean, my usual regular job before I got into the single cell community is very heavily in the EMR community, and we're the coordinating site for a number of quite large, we're among a combined set of coordinating sites for very large consortia that do take this clinical data and standardize it and produce it back for hundreds of research projects. And so I think the short answer to your question, Ian, is that there is been quite a bit of effort in this regard. And I think we're well past that for the level of data that is needed for those folks doing single cell analysis. And what I mean by that is there are 50 different institutions in each of 2 different large EHR coordinating consortium, the PCORI one, the CTSI ACT one. There are others. There's also the OHDSI one, but it's very epidemiologically oriented rather than patient record, individual oriented, that each have a set of robust used models that one can point to capture all this data associated with ontologies, all of that. It is a heavy lift to get from your own EMR to those standards, but it's so pervasive now that my short answer to your question is, you know, we do have methods and models and quite a lot of activity that has gotten past that issue that you raise, but it is not without a lot of effort and expense. And so one of the things that we've done in HubMap is to say, okay, I don't think we're prepared to just, you know, link these individual donors with their EMR, although we could do that. And some of our sites have an interest in that. Instead, we've taken more of an approach to say, okay, what does UNOS provide? And can we ingest everything that we're getting from UNOS, which looks very much like EMR information, but then store it in ways that are compatible with the EMR information. So that's a little bit about the answer to where the community is. It's gone quite a long distance already. And second, what we're doing specifically about that to take, you know, lab tests and vital signs and medications, diagnoses, all of these things, and putting them in with our donor metadata with those donors. We're right, we've collected now some of that, and we're right on the threshold of annotating our provenance of our donors with that information right at this time. So we don't have a lot to show for it yet, but we have a decent plan, I would say.
Thanks, that's a very optimistic overview. I mean, sorry, I'm not saying you're being optimistic. I mean, that's a good outlook.
Yeah, I think you know, to Anita's point, however, one of the things that I have experienced in the COVID-19 process, working on a totally separate thing, is that some of these processes are quite big and long and complicated. And there's often not a substitute for someone taking out a spreadsheet and saying, these are the 15 things I know I want to collect, and using the neural network inside someone's head to lay out the data that's appropriate for the particular circumstance by reading those records and laying out a spreadsheet that hits right on, you know, whether this lab value is positive or not, whether they've been in the ICU, several other things that are layers and layers of transactions if you try and pull them from the EMR. So I think that whole infrastructure is there and we should tap it as appropriate. But I also think we should be informed by it rather than assume that we're going to use it directly.
You know, I just wanted to say on this whole general topic, you know, there is a lot of work that has happened in the biopharmaceutical industry and standards around standardizing workflows and analyses and such as well. And I think it would be helpful to learn what some of these other groups are doing in terms of how do they manage their consensus process. Some of them have SOPs. And just to be informed so that this group doesn't have to reinvent the wheel all by itself and see, you know, take the best and then build on that for what's appropriate for this, this venue.
It certainly is an arena in which there are more standards than there are needed. So we should not make one.
Right. Exactly. Like, let's all learn from each other and try to coalesce around a common set is my dream.
So I realize we have about 30 minutes left in our session. And so I think two goals. One is to spend a few more minutes maybe talking through some of the questions we put forth under Breakout Session Part 2, as well as maybe at least try to fill out one of these blue boxes. I copied an extra one if we need more than one, but maybe spend 15 minutes on the green box, highlighting maybe what are our kind of top goals, where it sounds like establishing some best practices, but what exactly are those based on what we've discussed today? Maybe a place to start.
Just so I'm showing the right section of the document, do you mean this section?
Yep.
At this point, Sarah? Okay, great.
Yeah,
thank you.
So I don't know, Melissa or Kristin, if you have a thought of the best way to lead this part of the discussion, as far as kind of highlighting what are those best practices. I think that's what I'm getting from this second part of this conversation is to try to find areas where we can come together, whether it's defining specific clinical metadata. You know, we talked about maybe the use case, and if somebody wanted to highlight maybe from some of the COVID work that was presented this morning, if you're working on that, what was done to kind of unify kind of clinical metadata and how it was collected, or we can talk about some other context.
Sorry, I think somebody's not on mute. So I think it would be good if we make sure that we have some actionable steps documented. And I know we just had a long discussion. Sean, I assume you captured some of that here. I didn't have it up on the screen to watch. So, all right, I'm just having a look here. So we talked about some of the goals, establishing best practices, how to define the different metadata types and levels across the groups, how to work collaboratively with other groups who have been identified in the breakout sessions, such as the metadata schemas and ontologies, which it sounds like we were just touching on in the last conversation. How do we foster collaborations? Perhaps we could have some ideas on who to connect with there to have some actionable steps. Let's see. I think that what other main actions do you think we all could take coming out of this session? I think is something that we could focus on for the next half hour. Like, do you want to, for example, Jonathan, show the work that you've been doing and that's ahead of the curve of some of these discussions, and show where you're working collaboratively, and then how to bring in the other groups? That could be something as an example. The idea too here is that some of the people who are on this call will want to continue working together on these activities. That was one of the other requests that was made of us in setting up this session, in preparing for the session. So think about that. If you're not already involved, would you like to contribute to working in this space?
So Melissa, it's not, it may already be down in the notes, but I'd like to put down there as an action item the idea that, let's label it as the provenance discussion. It's got called various things. It's been mostly called that in this discussion today. It gets called other things in other places, but identifying a place where we can get to some consensus about that and a group that's looking at that. I don't know whether it's part of this group or whether it's somewhere else, but I think identifying that as a specific thing to get some answers to would be useful.
Okay, thank you. Anonymous Lemur, I assume that's you, Sean, for adding that.
That one's not me.
It's actually Sarah.
Oh, Sarah, thank you.
So I just want to get back to, I feel like the provenance discussion took us into an area where there's lots of existing solutions, efforts for collaboration. But I want to just circle us back a little bit and remember that, you know, across HubMap, HCA, various other efforts, that we have a sort of very global set of projects that are going on now. Samples being collected in different countries, different circumstances, not all with electronic health records. And some of those where collecting the donor information, the sample information, you know, some of those metadata fields are really key for us to be able to do these harmonized meta-analyses. And to get back, I guess, to Anita's point is there something that I guess the question is there something we need to solve here that we might be able to solve as a group? You know, can we suggest, based on analyses that are trying to do this and the missing data, are there a core set of metadata that we recommend being collected, perhaps? And is that missing? And I think the use cases of people doing the analyses are really going to inform that more than anything else across these data sets, because some of these projects are very well structured, collect a lot of information, and some of them have far, far less. And then I guess the question is there a set of harmonization needed? And then we didn't touch on it, but I do want to bring it up, which is a lot of the clinical metadata is often protected data. So how good are we at being able to share a lot of that as well? But I think just getting us back to, is there an issue that we need to solve? Is there missing data that people aren't collecting that really matters to the types of studies we're trying to do? Or is it in the harmonization of the way it's stored and extracted from databases? What are those two problems?
So you're touching on something that I've spent a fair amount of time looking at over, you know, I would say probably about 18 months or so. And there was some reference to somebody earlier who may have been doing something similar. But essentially what I was doing was looking at a large number of data sets, and this was in connection with what we're doing in NCI to do with the Cancer Data Service. And, you know, I don't know that I was necessarily bringing the topic of core elements myself or, to it. But that's almost a meme, and I think that is where a lot of our discussions end up going. I mean, it's interesting that you referred to missing data, because that's a hook that I would go into on this. Is that I've looked at data sets that have been, I've been taking the viewpoint, here's a load of sequence files that have been deposited, and I want to do some sequence analysis on them. I'm having to put myself in that scenario. It's not something I do day to day, but that's what our users are doing. But if I look at those files, and I've seen situations where there are 8 files from different samples from the same subject. So I want to know, okay, if I'm going to compare this BAM file with that BAM file, what's the difference? and I look at the data that has been provided, and I've encountered a number of cases where it's inadequate. In other words, there's something missing. And the reason something has become missing, as far as I can see in certain situations, is we've got databases that have said, these are the minimum fields you should provide. And it's not taking into account the uniqueness of the experimental design that was being conducted. And so, for example, one example was something that looked at organoids. And it wasn't capturing the two samples, you know, there were 4 samples coming from liver. They were paired, but it wasn't saying, and in fact, they were liver metastases, but there were 2 samples from one metastasis and there were 2 samples from the other metastasis. And those should have been being compared one with another. But the information to do that was missing because that wasn't part of the core metadata. So where I come to, so what is the minimal core data? What I can come down to, simply as I can put it, is that data which will allow me to distinguish this thing from this thing. But if you ask me what the fields are necessary to do that, it's not a constant set of fields. So if you ask me to enumerate the fields, I can't give you an answer that solves the problem.
Hey, this is Jonathan. Ian, I think that's a really insightful point. And, you know, our thinking has moved from defining those to almost the other end of the spectrum of taking the data that we can get and saying, okay, if you can organize and send to us whatever clinical data you have on this donor, we will annotate that, we'll associate with reference vocabularies, put the events in a referenceable model against this, and then you'll have the maximum chance of doing it. That raises the question more on standardization than on a field set of requirements, such that we're using reference terminologies so that we don't have a bunch of things that are totally incompatible with another. So we've focused much more on collecting as much as we can, associating them and annotating them into the sample, which actually at the donor level in our provenance graph, and some at the sample level, and then associating with reference terminology so that those analysis have the maximum chance of finding the thing that they're looking for to distinguish between different sets. That's a big commitment, and it's a bit different from what the community has done before, although we've seen the challenges associated with highly, you know, limited stuff in TCGA and other things. And so we are trying to solve that in that way. We'll see whether that's helpful. It depends on how well we really get the reference information right and make them, you know, data from one thing to the next cross-compatible in various ways. But I think your point is an excellent one. I feel like the efforts to say this is precisely what we're going to collect, that is the minimum standard, almost by definition just runs into this problem. And you can't ignore doing that, but at the end, it's also not anywhere near sufficient.
Yeah. I mean, I realized, just quick couple of points into what you're saying. I mean, and I think you're highlighting, in some ways, what I've just said is anathema, in that we want to standardize, and at the same time, I'm saying, well, everything's different and we've got to capture diversity. I would connect that by saying I think there's a standard way of capturing diversity, and that's where we need to shift our standard to be.
Yeah, extremely well said. Standardizing, collecting diversity. That's exactly what we're trying to do, or at least philosophically trying to do, by allowing minimal amount of structure, which almost entity attribute value type. What is this thing associated with? What is the value that you're collecting? What's the data associated with it? What is the content, whether that's a lab value or diagnosis or whatever it is? This is the way that parts of the clinical community have approached creating large analytic data sets also. It's not a new idea, but executing it in this community is somewhat new.
I think the other point you mentioned was that there perhaps are not data sets or systems that have this. I think there are some. We've been looking at them. I would say I've been looking at them in the examples I'm talking about. In many ways, they get a bad reputation amongst this community because they appear not to be standardized. And people look at these sources, and dbGaP would be one, EGA is another. But you can look at that and say, there's no standardization in those. Well, in fact, and it's true, that there's no harmonization of the data elements that are captured between dbGaP studies. But the one thing there is some harmonization about is the data dictionary, which everyone has to provide for that. So even if they were recording different things, they provide the description of what they did in standard format, which is, you know, the thing that I was referring to that you picked up on just there about the standard for recording diversity.
Yeah, I think there is an example of folks that are pretty far ahead on this amongst the Common Fund programs in the Kids First program. I don't know if there's folks here that are related to that, but they've definitely taken that attitude of send us everything you have and we'll organize it as much as we can. And they've made quite a bit of progress in that approach and making many useful things happen. So
although I think anyone that takes on the send us whatever you have and we'll organize it is creating themselves a lot of work.
It's not a panacea.
I want to jump in and say, I'm sorry, Jonathan, when you're done, if anybody else would like to jump in with comments, please, I'd like to open the floor to everybody as well. So I'm sorry, Jonathan, go ahead.
I am finished. Thank you.
Sorry, I didn't mean to talk over you. I just want to make sure we give everybody a chance to contribute to the conversation.
I just want to bring up, because I think it's a good point, is Ellen had put in the chat, and I see it's gone into the document, this notion of missing data, whether we're doing dissociated assays or spatial assays, what we care about in terms of mapping back to the body, organ, the tissue, is this collecting as much spatial information. And that's not the kind of thing that's available in medical records. It's the kind of thing that I think this group agrees is in the clinical data purview, because it's this upfront collection. So, you know, this might be an area to standardize some of that, and I'm sure it's done differently. It has to be done differently depending on whether we have a surgeon taking a sample, whether we have a team dissecting a tissue, depending on what country we're in, how fast or how slow something's collected. But I think it's an area of metadata that matters very much for us building a coordinate framework across the body. And I think Ellen's right, that it's one that nobody has really looked at standardizing. We're just trying to say, I've got this piece, I've got that. And I know the CCF, you know, the models for the CCF indicate that this can be very difficult depending on the tissue, but I think it's an area where it's a bit of a free-for-all right now. So I think Ellen's got a valid point there.
So this is Jonathan Himmelfarb. One thing that I think is important to consider, hasn't really been talked about much, is context of use for individual studies. So yes, we want to collect data that reflects the diversity of different purposes to which data can be put and meta-analyzed by different consortia and different studies. But each study often has a specific goal and resources available. And the kinds of data that are going to be collected really refer to what the context of use of that specific, at least initial use of the data is going to be. So I think our challenge often is to be looking in both directions at the same time. How do we collect the meaningful data for which a study is funded to obtain, and at the same time try to ensure that in the long run that data will have the most utility to a broader community. You know, I think there are a lot of challenges with that. I can, from the Kidney Precision Medicine Project perspective, I can tell you that, you know, we're very concrete. We're very much focused on building an atlas of the kidney, the human kidney, in health and in disease. And all of our funding is really related to that purpose. At the same time, we want our data to be as meaningful as possible and to be able to be coordinated with other data sets. So really one of the first things we did is we had a 4-day ontology workshop where we brought in leading oncologists, kidney anatomists, patients, clinicians, and asked where are the holes in all of the ontologies related to the kidney, work to fill those holes, and then create a kidney tissue atlas ontology, again, with sort of very broad community support. And then we had a controlled vocabulary workshop recently where we also had the kidney people that are in HCA and the kidney people in HubMap, along with KPMP, anatomists, again, patients, clinicians, cell biologists in the room for a couple of days to work on common vocabulary across all of these different consortia for anything related to the kidney. And then we work with all the other consortia that we know that are working in the kidney space in clinical studies or observational cohort studies, all the other NIH-funded studies, but also globally, to see if we can come up with common definitions for all of the terms that go into our data sets. So it's very concrete. It's all focused on the kidney, but at the same time, we're trying to be as collaborative as we can be so that the kinds of data that we generate can be used by as many different people as possible. But I think we also have to recognize the limitations that we all have based on what we're supposed to do, what we're funded to do. That really does define, to a certain extent, the kinds of data that we can collect.
So Jonathan, I mean, what I was referring to earlier, though I was describing it from the perspective of looking at the data as an analyst, what I was trying to do, and the approaches I was describing, we're trying to reach through to exactly the thing you're describing. It's essentially what I was trying to say in more ways is the people who know the information, which is the stuff that distinguishes these two samples and what could be derived from them, is that context that you're referring to about why, how were these collected. And I always feel a lot of the time, me as an analyst or pretending to be an analyst, we're trying to reach through back to the people you're describing through this middle layer of the data management, and sometimes the middle layer was stopping that reach through happening in a meaningful way.
So I just wanted to do a time check. We have about 10 minutes, so just make sure that we capture all the ideas for next steps.
So Melissa, I was just going to build on this. Now, the time is obviously entirely up to you how you wish to use. Towards the end of the document, though, I think we've got some blue boxes. There we go. Which, if at this point in the conversation you feel that there are even sketch ideas for collaborative opportunities that you would like other people to be aware of, so in other words, people outside this group, then being able to jot down your thoughts here is helpful, because we are going to extract those and put them into a sort of virtual poster gallery, just so everyone gets a little summary of what the groups have been talking about. And if it's not appropriate to do that, that's absolutely fine too. But I just wanted to highlight that in the last sort of 10-ish minutes.
I think Sarah has started to fill out each of those blocks. So maybe we want to try and flush some of those out.
Perfect.
Yes, and if we want another box, any more boxes too, flush those out. Thank you, Sarah, for getting us started here.
Yeah, no problem. I just took those two actions and kind of split them in. Obviously, I haven't kind of added moderators or note takers or members, so I encourage everyone to kind of fill in where they might like to participate if they'd like to represent. I know someone mentioned from the HCA they might be interested in representing the HCA in one of these groups, but I encourage you to fill them out yourself and whatnot. And then as far as what additional expertise, I think that'll be dependent maybe on who's in the group. It seems like there's a lot of expertise on both sides, but I think we may need to figure that out as time goes on, and then when the next meeting is, might require some follow-up as well.
So just a question for you, Andy. Are we supposed to fill out like this contact information as well from today's session, or is this like for what people want to do in the future?
No, I think the broad idea is to just allow people who were not in this group, because on any session there's a sort of fear of missing out because other groups are having interesting conversations as well. So I think anyone who wanted to stay in touch with this, it's not a commitment by any way, but it's an opportunity to say, actually, I'm interested in this conversation and I was part of it and I would be interested in following up. So.
Thank you.
Andy, can I ask a follow-up to that? Are you guys capturing who was a participant in this group discussion today, or is it be good for us to just write our names down so we know who was involved?
So, in fact, I did grab a screenshot of our participant list, but I would encourage everyone to put their names down, because it's also possible. It looks all right in this group, but sometimes people's usernames are R21 or something like that. Actually, R21 is an odd choice, but
so I started a list just below the last of the blue boxes if people want to add their own names. I only added my own so far.
Yes, I would encourage everyone to do that, just so we know that you were interested.
So just to clarify, these are what we're listing right now, I think, are participants in this session, opposed to if you're interested in one of these topics, to also maybe add your name to the blue topic box as well, or your email.
Yes.
Is name enough because we have email contacts for everyone, or people should add their email?
No, I would imagine name will be fine. Yes, because everyone is registered for the event, so we will have your details.
All right, so I just see we have about 4 minutes, and I'm sure everybody wants a break. So we have 2 blue boxes, which are going to be what everybody else sees when they look at our outcomes. So we have one is about establishing best practices for defining core clinical metadata across groups or consortia, and the details about that. The other topic we have here is identifying the consensus on provenance.
Which can certainly be renamed. I just focused on the context.
Well, and it has more context here of, as part of the clinical metadata, naming and establishing the conventions of limiting, that might be a typo, of including the semantics, including the semantics that we need for the sample name.
I mean, that's a relatively, that's something we should certainly address. I mean, I would expand it a little, Melissa, to make it a little bit clearer and say recording provenance. If you could scroll back up a little bit. Yeah, identifying consensus on recording provenance. Because it sounds like we're, you know, let's establish what the provenance should be. That's not what we're talking about. We're talking about how you would record it. Different provenances.
I don't know why it keeps going to bold. Okay. All right. Is there another major takeaway topic that we want to post out?
I think there, Ian and I had this sort of back and forth about whether establishing the common clinical metadata, or if it's about essentially addressing the context of a data set. And somehow that either should be represented in the first box as the context, maximizing the context of a data set really is the goal. I think there's a specific goal there, and we heard that from Anita and others. So maximizing that context, I think, may even be another whole box.
I would,
or maybe it's in that one.
I would tend to agree that, I mean, if we could see what the first one was again, I've got it here. But I tend to agree that we should consider it separately, but later maybe merge it, is my thought about it. Because I definitely see the overlap.
Yeah, I guess then putting it in there in that blue box, maximizing the context, what we know about a data set, I think is really the specific goal. In other words, there's no intrinsic value in standardizing. There's a goal of maximizing the ability to understand context. And so we need to constantly keep that in mind.
I'm sorry, I was typing something else and trying to edit. Could you just say that again, please, what the title of the—
probably not another box, actually.
Yeah.
I think—
Not another box.
No, I think that one was fine. I think it was in the other one, actually, maximizing the context of a given data set. It says problem we think we can solve.
I think the key word is context.
We're not really about solving standards. We're really about solving the analytic context, the context of the data.
The context of the study is what I would suggest.
It's a context of so many things, but yeah.
I mean, I have a concern once we end up talking about these terms like context and provenance, which are rather abstract, and I wonder whether they always connect with what the real use case is. Because the question it's come up, the question about time and time course and samples over time has been covered, but that is covered within what we're talking about in terms of this context. Time is one of the place on a study is one of the contexts that's important. The context is this horribly abstract term, just as provenance means, this is what was done to this specimen. And we should, never lose sight of those connections.
Jonathan, maybe you're typing. All right, well, you feel free to edit this. All right. Well, it looks like we're after the top of the hour here. So any last thoughts and points we want to capture before we wrap up? I think we have a 45-minute break now for, well, if you're in the Eastern time zone, it's lunchtime, but could be some of you are calling from other places, I know. So whatever the appropriate meal is at this point in your day.
This is Jonathan Himmelfarb again. One last minute thought is that the informed consents will dictate sometimes exactly what kinds of metadata, what kinds of data can be collected or in what fashion they can be collected. So thinking about this has to go all the way back to, in each study, to sort of the initial planning of the study. If you get it wrong in the informed consents, it's very hard to fix it after that. And
good point.
On the tension between protected health information and PII and maximum utility of information, but that's very important too.
All right, and with that, I invite everyone to go and stretch their legs. You will be able to see from the navigator that on East Coast time at 1:45, we're coming back into plenary briefly, and then we're into our next round of breakout groups. So I may well see some of you in my next group, which is metadata, schemas, and ontologies, and if not, then I hope you have a great time in your other groups.